It's a real privilege to have Tomas Paus here, and I don't know if everybody knows him, but he's a world famous guy who's also very nice, and it doesn't always go together, but it does in his case. And it was interesting for me to hear the story. I mean, your story is kind of like a vacation. You know, there's Europe, there's England, there's Canada, and there's a little bit of Santa Fe. You know, you're not going to the dumb parts of the United States. You know, so and you're some sort of professor of important things in Toronto. I'm sorry I didn't look at your bio because I wasn't expecting to stand up here. But trained at the famous M&I Institute, which if you're in neurology, and I presume for you guys in psychology, it's amazing. And my daughter's going to McGill, and we walk past the M&I Institute when I drop her off, and it's like, wow, it's the history of neurology. Have you passed by Penfield's places? It's just incredible. So if you have any time to kind of sequester Tomas on your own, you really should get some time to chat with him, because it's really nice to find out how your career's evolved, and you're doing just amazing things that we have all been following. So thank you very much for coming. And tell us what you are a professor of. There's a formal great
chair in population neuroscience.
Very nice. Thank you.
Anyway, not anymore. Thanks, John. It's really great to be back and you know, see, of course, old friends and my beginning in Santa Fe with Alan Goldberg. It's really, that's the reason why I keep coming back. And of course, the mind that I've had now an association with your institution for quite a few years, and you're doing some really interesting work here, obviously, as well. What I will do today will be somewhat different from, certainly different from my talk here, I don't know, 4 years ago or so, and very different from the talk I just gave in psychology, which is more about the population neuroscience, life studies, and how we are trying to understand the influence of genes and environment on brain development. I selected one particular topic that is very close to my heart, and that's really white matter. White matter that you have been always, I'm sure you've been doing a lot of work along those lines on white matter and connectivity. And I will try to convince you today that very often we are ignoring another very important function of white matter, and that's what I'll be talking about, the function being the transport function. So the layout of today's talk is really the introduction of terms. Then I will explain to you why we can think of white matter as both wires and the roads, and then I'll get into two sets of studies that started with human work, but then progressed through animal models and tissue cultures to show you the relevance and where we are, why we are thinking about white matter as a transport system. So connectivity. There are so many connectivities, and it's in a way unfortunate that imaging has decided to use the term connectivity and apply it to everything, pretty much, right? It's a metaphor. In the context of our work, connectivity is but a metaphor. It starts, of course, with functional connectivity, and as we have been doing, if you are with MEG, EEG, very often it's about inter-regional correlations of time series, and we call it functional connectivity. But obviously, it's just that. It's a correlation, sometimes calculated in a more fancy way than other times, but that's what it is. Things go up and down together. There is no causality, of course, in those relationships. Then we can try and inject some form of causality into connectivity, and quite often we talk about effective connectivity. The idea is that yes, there is this region A that is driving region B. And that's very difficult to establish, at least in vivo with human studies. Certainly very difficult for fMRI that doesn't have the temporal resolution as EEG and MEG, where in those electrophysiological time series there is a hope for what is called sometimes Granger's causality, a precedence in time of one region, really the change in one region driving the change in another region. The way that I've approached it about more than 10 years ago was combining transcranial magnetic stimulation with brain imaging, an idea, a very simple idea that goes back to the 30s, when that was before the tracer studies, when people like neurophysiologists like McCullough would put a little bit of strychnine on exposed cortex of a monkey, and then follow the spread of the local seizures, right? So that was called neuronography. And that was really a way in which they really mapped out effective connectivity in this way. And that's what we did really without the seizure part, of course, with brain stimulation. We would stimulate focally one part of the brain, and with that measure the distal response to that stimulation. And then we have, of course, structural connectivity. Now, structural connectivity has many faces. We can think of structural properties of white matter, starting with simply a volume, and I'll give you some examples of that. Or we can measure microstructural properties of white matter. Of course, with DTR being the most popular method for measuring structural properties of white matter, you also read a lot about myelination. Well, I don't believe it, because DTI and DTI-derived properties are not simply reflecting one of structural properties such as myelination. It's a composite, of course, of many different things going on there. Myelin thickness, axonal diameter, orientation of the fibers, their packing, etc., etc. There are very few in vivo imaging techniques that have the level of specificity that allows us to make a link between what we measure and a particular neurobiological or cellular compartment. One of the best ones for myelin is myelin water fraction. So that comes very close, I think, to measuring myelin content or myelin concentration. Another one that I will use just briefly in my talk is magnetization transfer ratio, MTR. Not as good as myelin water fraction, but again, it's telling us something about the relative content of myelin, at least in the typically developing children. But this is connectivity. That's the only time that I would feel comfortable to say neural connectivity. Because I inject a tracer here, wait for a few days, the tracer is picked up by the cells and transported either in anterograde or retrograde direction to one synapse only. And then I can pick, I have to kill, unfortunately, the animal, and then I can see where that tracer got. And obviously, it could get there only via axonal transport. It could get there only if there was a population of neurons here that picked up the tracer, and those axons went here and there. That's true connectivity. So tractography is not connectivity. It's a model, a mathematical model of the best possible representation of water molecules moving in a particular direction. But it's not connectivity, certainly not point-to-point axonal connectivity. This is connectivity. Okay? So that's some of the introduction of terms. Another interesting thing when you look at work by Valentino Breitenberg and his colleagues, I'm blocking her first name now, Schütz. They did some modeling based on some basic quantitative anatomy, and they discovered that in fact the majority of long distance, of course, strands, which are the ones that we typically study with ETI, the percentage of those strands to all white matter is minuscule, 8%, something like that. So when we study with DTI connectivity of those large fiber tracts, we have to keep in mind that we are studying 8% of white matter, because most of the strands are either in the U fibers that are the local connections, 3 to 30 millimeter long, but the majority are really the short axons within the cortex, the intracortical fibers that are less than 3 millimeter long. So imagine that you are making some inferences about properties of white matter based on the 8%, and those properties may be the same, or they may be also different from the short axons. And of course, you can imagine how important those short axons are for local intracortical computations for the modules that we have within the cortex. So let's keep that in mind. And And now, of course, for me, what is important that there is, and that's disputed sometimes, it's not a perfect relationship, but usually the longer the axon, the thicker it is. And that, as you will see in the next few minutes, may be an important property. So these are, again, there will be a variation, but most likely these axons are thicker than these axons, and you will see in a minute why I emphasize that. Right? Now, the axons, and you will see a lot of this in the next 30 minutes or so. The axons in the human brain, most of them are myelinated. So there is the layer of myelin here, and here is the axon. Two different cells, right? Because the myelin is generated by oligodendrocytes that are wrapping the myelin sheath around. So that's one cell, glial cell, and then there is the axon, that's the neuron. Now the ratio, it turns out that the ratio between the axon diameter and fiber diameter, so-called G ratio, is about 0.6. And you will see in a minute that that ratio varies actually with the size of the axons, with the axon diameter. So let me move now into the wires and rods. So that's really, typically that's how we think of white matter, right? The basic physiology, conduction of action potentials, communication, transfer of information, electrical impulses. That's electrical grid in Quebec. Now, we should be thinking of white matter and axons also as rods. Now, that's an interchange also in Montreal. Anyone notices something weird about this set of roads? There are no cars, right? Because it's still being built. They don't go anywhere. But the idea is, of course, and I'll tell you in a minute why I think it's very important to think of white matter and to think about axons also as roads that are transporting cargoes. And here is the reason. Whatever you need at the synapse, many of those things are, of course, made in the cell body, because that's where the synthesis of proteins is taking place. And, of course, therefore, all that material, all those organelles, all those cargoes have to get from here down to here. And they get there through a wonderful system of cytoskeleton, a system that consists of microtubules and the motor proteins that are marching on top of those microtubules. So one motor protein comes from a family of kinesins. Kinesins are motor proteins that are moving cargoes in anterograde direction from the cell body to the synapse. Dynein is a motor protein that moves cargoes such as growth factors from the synapse in retrograde direction to the cell body. And you can see that this kinesin protein has basically legs here that are burning ATP, and that drives the movement, walking of the protein along the microtubule, and depending on the electric charge, it would be moving, of course, either this direction or that direction. And here it has hands that are going to grab those cargoes, hold onto them until they reach a certain destination when they offload those cargoes. And some of those cargoes are offloaded, in fact, on the way, because some of them may be needed at the Ranvier, how do you call it? Node of Ranvier, thank you. And before they get to the synapse. So it's a very sophisticated machinery that needs to know what to do on, where to deposit it, move it this direction or that direction, right? And here is what the cargoes are. So, of course, they are, as I said, synaptic vesicle precursors, receptors, transporters, enzymes, everything you need for neurotransmission. You need to get it down there to the synapse. But also for cell metabolism, mitochondria being the most important one. And for the cell survival, it's usually in the retrograde direction. So if you don't suck in those growth factors from the extracellular space and load them up and move them up here, your neurons die. And there is a lot of work now that is going on about neurodegenerative disorders that suggests that it's the axonal transport that is in part responsible for what they call dying back degeneration. It starts actually here, and that's where then that causes the death of the neuron. Okay, now I will get to the point when I'll explain to you why I'm interested in the transport system. And it really starts with one of our population studies, the Saguenay Youth study that we started more than 10 years ago back in Quebec. That was really the primary purpose was looking at the relationship between prenatal exposure to maternal cigarette smoking as an example of adverse intrauterine environment and brain development in adolescence, and of course brain and behavior in adolescence, but also in fact cardiovascular and metabolic health. And I won't be talking about that at all here today, but I will show you what motivated the work on white matter and the transport. And that slide is really the beginning here that shows in a subsample, the first 300 or so individuals, shows that during adolescence, between the age of 12 and 18, white matter volume, absolute or relative adjusted for brain size, does not change much in girls, but it really goes up, the volume goes up in boys, right? Now I can tell you that it's not because there was a large increase in volume in girls before the age of 12. It's really the case that even when we look at other studies, there is a more gradual increase in white matter volume in girls, and then there is that accelerated increase in the volume in boys. Now we'll get to why that is and what it might mean. Well, I don't know that this part. Now, obviously, one big difference between male and female puberty is testosterone. So we ask the question: is that increase related to an increase in testosterone levels during male adolescence? Of course, age and testosterone correlate with each other very strongly, so we took a different approach to try to get potentially a causal relationship between testosterone and that increase. And that approach is simply looking at gene-environment interaction, where now the environment is testosterone, and the gene is a gene that is, of course, important for, or the protein that is very important for translating the effect of testosterone on the cell, and that's androgen receptor. Now, androgen receptor comes in many forms, different genetic variants of this gene. One type of variant, which is functionally relevant, is the number of CAG triplets in exon 1 of the androgen receptor gene. If you have extreme number of triplets, 42 and more, you have Kennedy's disease. That's a disease that is characterized by degeneration. It's also called supranuclear bulbar. Can you say?
I think it's supranuclear palsy.
Yeah, right. But so it affects the axons, but also it's associated with hypogonadism, right? Now, of course, even in the normal kids, we have a range, and what we can do is we can genotype everyone, count the number of those CAG triplets, and then have a very simple prediction. If you have more above median number of CAG triplets, we'll say you have an inefficient androgen receptor, and the ones below the median have an efficient androgen receptor. Now, the prediction is very simple. If you have the efficient version, testosterone should be, if it's really about testosterone and not something else, testosterone should have much stronger relationship with white matter than in those who have the inefficient version. And that's what we see here. So these are the ones with the efficient version, short androgen receptor gene. These are the ones with the inefficient version. And you can see that testosterone predicts different, explains different amount of variance in the relative volume of white matter. So that's good. So we know, at least in part, that increase in the white matter volume during male adolescence has something to do with testosterone. But then the question is, what is it that is increasing, right? And I hinted at it in my introduction.
we have at least two components here. We have an axon and we have the myelin. So typically when, before our studies, when we are talking about white matter and development, we always think about myelin. We tended to ignore the possibility that axon may also have something to do with that. Now, I told you that there is at least one, or there are a few MRI sequences that allow us to get an index of myelination, and the one that we used in this particular study is called magnetization transfer ratio, MTR. So if it was about myelin, then what you would expect is that MTR would go up as a function of age or as a function of testosterone. And what we see is that it goes down, right? And it goes down more in those males with the more efficient androgen receptor. So what's going on here? Are they demyelinating our poor boys? Probably not, right? So, and here is the answer, I think. At least that was the speculation the explanation that we gave in our first paper. This is electron micrograph from the monkey corpus callosum, and you can see that axons come in different sizes and they have different thickness of the myelin sheath. You can see also, and that's not a different amplification. This is the same amplification on both sides. There are some gigantic axons, right? Now imagine, that's a caricature, but just so that you know what I'm thinking about, imagine that what happens during male puberty, you go from this to that, right? What that means for MRI, for MTR, there is very little space for myelin. It's now, relatively speaking, that 1 millimeter or 2 millimeter voxel that we use to measure MTR is filled with the axon. And so you get a relative decrease or relatively less myelin. And that was our explanation for that apparent decrease in the myelin index. It's only because it's relative. Of course, we don't think that they are losing myelin. But when you go, if your imaging voxel was, if it were to contain only this, obviously there is less myelin here than there is here. So that's just simply an issue of space. But it's also true, and that's why I was talking about G ratio, it's also true that as the axon diameter increases, the G ratio, which reflects the relative thickness of myelin sheath, the G ratio goes up, meaning that relatively speaking, the myelin thickness is less. There is less thick, right, for a given size of the axon. So you have two mechanisms by which you may be apparently losing myelin, because there is not more space for it, and because the large axons have actually less, relatively speaking, less myelin. Now, I told you that there is a relationship between length of the axon and, in fact, G ratio and its thickness. And that's why we went on to look at one particular tract that is actually the longest one in our brains. Anyone can guess which one is the longest tract in our brain? Cortico-spinal, right? From the motor cortex down to the cervical, let's say, spinal cord, right? Now, it turns out, and here we have an axial section through the internal capsule. That's the thalamus, putamen here. That's the internal capsule here. And it turns out if you use myelin stain in postmortem brains, there is a little patch here in the internal capsule that looks paler than, it's more pale than the neighboring white matter. So it looks less myelinated. And that's exactly where the cortico-spinal tract passes through. So that was, again, a hint that we may be right here, that in fact, because the cortico-spinal tract consists of many large axons, there is actually less myelin compared with the more, perhaps smaller and more compact fibers that are passing just in the vicinity of that patch. So we went back to our data, again in the Salmon-AU study, and look at the average of gray matter. So that's a gray matter probabilistic map from maybe 409 participants. And we look at the internal capsule here, and there is a pale region here, and it's pale. In fact, it's misclassification of white matter, because that's all white matter, right, in the internal capsule. It's misclassification of the white matter here as gray matter. See, this is the probabilistic map of, in fact, gray matter. That's why you see thalamus here, putamen, caudate, etc. But there is a little bit of gray matter in the internal capsule. Obviously, it's not gray matter. It's simply that, again, the white matter is less white. And the algorithm is fooled by that, and it says, oh, you've got gray matter here. No, it's, again, the cortico-spinal tract passing through. So we extract that signal here, and we see that that signal, that paleness, if you like, is increasing with age in males, and it doesn't change at all in females. So again, consistent with the idea that something is going on with that axonal growth. Now, and that's testosterone is explaining again a big part of it. Now, the last little piece from the human studies. We then went, and as John said, we traveled a little bit, so I traveled to England, lived there for 5 years, and one benefit was that we had a nice 7 Tesla scanner, and so we repeated those studies now at 7 Tesla in males, a small sample, but that allowed us to really zoom in. We had the resolution, we had the signal. Now to quantify the properties of this patch, again, this is now single subject. You can see the patch even in the single subject. Quantify properties, MTR and T2 relaxation time from this little patch and compare it with the neighboring axons from the internal capsule. And you can see that the difference is huge. So here we have MTR in this patch of CST, cortico-spinal tract, low MTR, as we would predict, because it's relative, right? So if the axons are big, there is less myelin, compared with just adjacent white matter. And that's the same thing now for T2. As we would expect, the T2 relaxation time is longer in that part compared with, again, the white matter adjacent to that putative cortico-spinal tract next door. So, of course, that was all nice, but we did not measure the axon, and that's what I wanted to do, and that's why we went on with a new postdoc in the lab, and we went to the animal model. So let me tell you about that. So what we did, we went to the rat, about 2 months old, so young adult here, and we went to the splenium, in this case splenium of the Corpus callosum, and measured the axon diameter and fiber diameter in 50 axons per sample, so close to 400, I think, axons per animal. Then we have the measures of the axonal diameter and also the measure of the myelin thickness. And then, so from those 400 measurements per animal, we can get the median. Obviously, this is a very good estimate because it's based on 400 measurements. And we can look at the differences between males and females. And the differences are highly significant, confirming that yes, indeed, the axon diameter is thicker in our male rats compared with the female rats. And that the G ratio is behaving again the same way, both in males and females. It's increasing as a function of axon diameter, but also it's higher in males compared with females. Unlike the Sagonae study where we needed about 200 males, here we have 6, and it's significant, right? So it's wonderful. Animal studies, when they work nicely. They give you p-values with very small sample size because you have such a clean data set, right? Now, of course, so that indirectly, that sex difference is, again, confirming that yes, androgens may have something to do with that. But in this case, we could really push it a little bit further and ask, well, what happens if the androgens disappear? And we make them disappear very simply by castrating the males at weaning. And so here we have the effect of castration. The castrated male, the axon, the diameter is different from that of an intact male. And it's not significantly different, but that's a sample size issue here, right? I mean, there is a trend, so they are probably somewhere in between male and female, but in this case, statistically speaking, not different from females. And we see something very similar for G ratio. Again, the G ratio of the castrated male is very similar to that of a female. So that really confirms now that what we were assuming is going on in terms of the axon diameter and G ratio. Certainly it is going on in the rat in terms of sex differences, and that removing testosterone just before the onset of puberty is reducing greatly that sex difference. Now, of course, castration is not going to remove only testosterone, other hormones, the hormonal environment is different, so that again still leaves out the possibility that other than testosterone effects may be explaining the differences. Now, there was one more interesting observation, and that was the distribution of axons of different caliber. As you can see in males, there is this tail of large axons. Not many, but they are there. And that tail, that uneven distribution of axons is eliminated by castration. You can see that both females and the castrated males have pretty much symmetrical distribution of the axons. They don't happen to have those few large axons. So it's not about moving the whole distribution to the right. That would have been the case, that all axons are a little bit thicker. That doesn't seem to be the case. It seems that testosterone is influencing the probability, the percentage of really large axons, right? So that's what this plot is showing us. So let's go now. I know as a psychologist you don't care about anatomy, which you should, but I'll give you now the punchline, because of course I'm doing it because I am interested in the consequences of all this. The anatomy is at the end what is the basis, the foundation for all the functional properties that we have. So, and of course I told you about transport, and the obvious question is there a relationship between the thickness of the axon and axonal transport? Or another question, is testosterone influencing axonal transport? So that's why I'll come back now to the question of axonal transport. Now, there is one study that has been done. That's just to show that, yes, of course, the thicker the axon, the more microtubules you have. So there are more rows, if you like, right, in the axon, if you have a thicker axon, because that's what it is. One really nice study that we are now, and I don't have data for that yet, but I'll tell you what we are doing. Has been inspired by what was done in Nikos Lokotetis' lab in the monkey with manganese. Some of you might have heard of manganese imaging. Manganese is really a contrast agent that is used sometimes just simply to enhance contrast in animal studies, but it's toxic, so neurotoxic, so you would not want to use it in humans, obviously. But what is nice about manganese is that if you put a little bit of manganese, in this case, into the eye, the retinal cells would start picking it up, and essentially it flows in the same way as calcium would flow in. So it's really stimulated. The uptake of manganese by the cell is stimulated by glutamate, right? By excitatory neurotransmission. It enters through, I believe, the same channels, the calcium channels. Now, once it enters, it gets into the vesicles, and the vesicles are transported in anterograde direction to the next station, right? And then even to the next station. So now you have, in fact, a tracer. Manganese is used, can be used, as an in vivo tracer of neuroconnectivity, and people have used it this way. Now, what I'm showing here is something that was not, I believe, the primary aim of this paper, but it turns out, for those who work in the visual system, that we have the axons that go into the lateral geniculate nucleus come in two different sizes, small, parvocellular, and large, magnocellular. And they end in different parts of the geniculate nucleus, LGN here. The magnocellular end in this part, the parvocellular end in this part. And so what he was able to do is to simply plot the signal change as a function of time separately for the big cells and the small cells. And you can see that manganese got to the destination faster for the big cells compared with the small cells. So here is a nice potential model to study, in fact, axonal transport in vivo. Now, we are doing it with mouse, and some of you might have wondered what was that stuff in the middle of my first slide, and that's olfactory system. It's a painting by Golgi, and the reason I put it there is that you can use the olfactory system as a nice model for axonal transport. You can do nasal lavage in a mouse of manganese. You just drop a little bit of manganese into their nose, and the olfactory cells pick it up, and then you can do the scanning of the olfactory bulb and measure how much manganese got there. And that's exactly what we are doing with gonadectomy in mouse, but the data are not finished yet, so I won't be telling you about this. But that's why the manganese in vivo as a tracer is very useful for studying axonal transport and whatever may be influencing the axonal transport. What I will show you is the data that we obtained in vitro. Data that for that we used cultures of sympathetic neurons. We would play those sympathetic neurons in one corner here, right? They grow their axons. Once the axons are in place, then we would add synthetic testosterone called mibellarone. So that's synthetic testosterone that cannot be converted to estrogen, right? That's why we are using this particular version. So it's really, I know now that whatever effect I will see, it's really about testosterone. And we use it at 2 doses, sort of medium and large dose of testosterone. And of course, what we are interested in is asking, okay, so is the axonal transport in those sympathetic neurons, its speed, its amount, is it affected by adding the amount of, small amount of testosterone? And the way that we measure axonal transport is we add to the culture, at a certain point, we add wheat germ agglutinin, which is a tracer that has been used in the past also in studies of the sort that I showed you with the monkey. But now what happens is that wheat germ agglutinin is taken up by the cell, loaded into the vesicle, and then transported using the same mechanism that I showed you, both in anterograde and retrograde direction along those axons. And it looks like this. This is a vesicle loaded with the fluorescent wheat germ agglutinin. It's going along an axon here, and I can easily now, it disappeared a little bit from the focus, it reappears now, and it continues. And I can measure 100 of those vesicles, and therefore I can really, I see the transport. You know, for MRI people like me, now I can see something real. It's not only math behind a reconstructed image. This is not a reconstructed image. It's really fluorescent light marching along the axon. Amazing to me. But anyway, that's why I like this movie. Now, so has it changed? And we measured the effect of testosterone after 6 hours, 24 hours, and 48 hours at the 2 different concentrations. Now, the interesting thing is, compared with control, there is not that much of anterograde. I'm plotting here the ratio of anterograde to retrograde transport. So there is not that much transport moving in anterograde direction, but adding testosterone brings it even lower. So something is happening here. Testosterone short term is going to decrease even further movement of vesicles in anterograde directions. Okay? Now, after 24 hours, everything goes, anterograde transport goes up in all conditions, and there is not much of a difference between the control and tester conditions. But 48 hours later, anterograde starts going down in the control condition, and that's certainly not the case for the testosterone, especially for the high dose of testosterone. So now we have this very interesting situation. After the first 6 hours, imagine that you are an adolescent boy. You have a surge of testosterone, and this is true, and that's happening in his brain. Then axonal transport moving in anterograde direction is attenuated. And nothing changes, the testosterone remains the same. 48 hours, it overshoots, right? And now imagine all the consequences of that in terms of the availability of neurotransmission-related machinery and the possible imbalance that may be brought about by this fluctuation in the levels of testosterone. Now, one more thing, we can also measure for how long the vesicle, or the motor protein, is going to hold onto that vesicle. That's what we call distance of the run, right? And that's, you see, we are tracking the vesicle, it's moving, then it stopped for a little while, you saw that there, and now it's moving, like that. And the maximum run length before dumping that vesicle is greatly increased even after 24 and after 48 hours by the addition of testosterone. So what may be going on is that short term, the 6-hour effect, may be non-genomic. We know that there are androgen receptors also at the membrane. They may be mediating the direct non-genomic effects that would be influencing really the cascades that have to do with phosphorylation of proteins that are involved in holding onto the vesicles here. But the long-term effects are most likely genomic, and it's possible that once the testosterone gets into the cell, I mean, that's the usual mechanism of the genomic action, it gets into the cell, it binds to the androgen receptor then, and that complex enters the nucleus and starts increases gene expression of particular genes. In this case, we would argue most likely it increased the expression of kinesin. So now, after 48 hours, you have more of those motor proteins that are moving cargoes in the anterograde direction. But obviously, all those molecular mechanisms must be verified. So what I try to do today is to tell you that, yes, we can go actually from population to experimental neuroscience. We can get some interesting inspirations and have some informed guesses about what's going on with our in vivo data that we collect in this case, typically developing adolescents. But then we do need to go into the model and check out some of those hypotheses, either in this case electron microscopy and then in tissue culture. And as I said, we are now doing the last part, looking at differences related to gonadectomy and manganese axonal transport. And really, I think that, as I said, just try to think now about that male brain during adolescence, and you realize that it's no benefit that their white matter is growing more than the girls' one. I think if anything, it might be a risk factor that perhaps explain, at least in part, why disorders such as schizophrenia begins 5 years earlier in men than in women. And that's what we know. And that may be one of the reasons for that. And I'll stop here by acknowledging my